Welcome to our virtual space, where thought leaders who, in a variety of ways, have committed themselves to improving our lives share their work, perspectives on current affairs, and what brought them to where they are today. My name is Rob Liu, and this is The Exchange. So, Matt, thank you so much for joining us on The Exchange. We are in incredibly interesting times with the COVID-19 pandemic that we're all experiencing. And so I'm really pleased that you were willing to have this virtual at home interview, because from listening to you at a number of faculty meetings and a number of gatherings, I know that there's some very interesting things that you have to say that I think the rest of the world would really benefit from. But before we get to that, I want to give our listeners and viewers sort of a sense of who Matt Meselson is. And there's no question that, your work, sort of most famously with Franklin Stahl, where the two of you together did what is commonly called the most beautiful experiment in biology. I'm sure that you've been asked this a lot, but I'd love to have you perhaps spend a minute or two telling us how this experiment came to be, and in particular, the breakthrough in our understanding of DNA and genetics that came from this experiment.
Well, it really began with a lecture at Caltech, where I was a graduate student, by Jacques Monod. Jacques Monod and Jacob, Francois Jacob, as you know, got the Nobel Prize for their work on how genes in bacteria are controlled by a repressor. Well, Jacques came to Caltech at a time when the question was, when you induce the appearance of the enzyme beta-galactosidase by presenting bacterial cells with either lactose, the substrate, or a synthetic chemical that's more often used to induce, is that enzyme already lurking inside of the cells and activated by some minor change, addition of a methyl group or something? Or is it synthesized de novo? So that was the big question. And Jacques proposed that a change in the Donin equilibrium could be exploited to see which it was. Because if you make a lot of a new protein, then the osmotic pressure inside the cell goes up, water should pour in, and maybe you could detect the swelling. I'm sitting in the audience thinking, this is the world's worst idea. Because, first of all, what do we really know about the bacterial membrane? Does it have holes in it? Is it going to respond in that way by building up hydrostatic? It's a terrible experiment. And I had been taking a course, I was the last student of Linus Pauling. I was his last graduate student at Caltech. And I'd been taking a course from Linus on the nature of the chemical bond, and that we were thinking about deuterium that very week and what the strength of a deuterium bond would be instead of a hydrogen bond. So I'm thinking about heavy isotopes. And I was in chemistry, structural chemistry, in order to become a biologist. But I thought I should learn about molecular structure first. That's why I was taking that course. That's why I was working for Pauling. And so I had the idea that the right way to do the experiment that Jacques Monod wanted done was to grow the bacteria in deuterium. So all the replaceable hydrogens would now be heavy hydrogens. All the covalent bonded ones would stay there, even if you change the bacteria now to ordinary water. So the idea was grow them in heavy water, add the inducer, quickly move them to light water medium. And now, if the enzyme was synthesized after you added the inducer, but wasn't there already lurking, if it was synthesized after, then it would be made all of deuterium, no hydrogen. So that was the experiment. And to tell whether the enzyme was heavy or light, I had the naive idea that you centrifuge it in a solution of sucrose that was just in between the calculated densities for the heavy and light beta-galactosidase. Then at this point, I decided I would try to visit Max Delbruck. He was in biology division, but he was a physicist. And I wanted to get to apply physics to biology. But Max had a fearsome reputation, undeserved, but he could be very short if he was hearing nonsense. So I went to see him in his little office. And the first thing he said is, what do you think about these two papers by Watson and Crick? This was 1953. They had just come out. I said, I've never heard of them. So he jumped out of his chair and grabbed a little heap. Jim had sent him a stack of both reprints of both of those Nature papers, and Max threw them at me. And they fluttered down onto the ground, and he shouted at me, get out and don't come back till you've read them. So when I went back, he told me that he didn't think that their idea for DNA replication could be right. Because think about it, here you have this double helix, and you have to get the two chains apart. And unless you break them, that means you have to unwind the whole long thing. And you can calculate from simple hydrodynamic equations that for a long DNA molecule, it's an impossible amount of work. The other way is you could take the two arms and wind them. That's even worse. So Max had a different idea, which is to make breaks around single bonds. The chains you can rotate around a single bond. And so if you break one of the chains, it doesn't matter which one, you can swivel around the remaining chain because it has single bonds in it, and then seal it back up again. Or seal it up differently to other places. It's complicated, and I won't try to explain it. Max chose the other alternative, and that's wrong. And it would have resulted in a chain of DNA that was part made at one time and part made in the previous generation. We know now, of course, that breaks are the answer, all right, but it's done by isomerase. Mm-hmm. Anyway, so immediately I thought of my old beta-galactosidase idea that we would use density to determine whether the DNA had been, whether there were old chains and new chains that retained their identity, or whether they got all mixed up by Max's breakage scheme, or maybe even by some mechanism that nobody had the temerity to approve. That you have a DNA molecule, the two chains stay always together, and somehow they make another double helix over here. There was a proposal for how to do that from a physicist based on resonance theory that was no good at all. Anyway, then it's time to go to Cold Spring Harbor. Jim Watson wanted me to go and do an experiment for him there, but pretend to be a teaching assistant so I'd get paid.
Okay.
But I wasn't a teaching assistant. I was doing an experiment. Nevertheless, I used to join Jim. This was a great summer, 1954. Jim was there. Francis Crick was there. Sidney Brenner was there. Rosalind Franklin was there. A whole bunch of people were there. And so one day they were planning the course for the coming week. And Jim went over to the window. And he pointed out the window and he said, you see that guy sitting down there on the grass across the street? His name is Frank Stahl, and he thinks an awful lot of himself. Why don't we give him a really difficult experiment to do? See if he can do it. We'll give him the Hershey Chase experiment to do, all in one afternoon, just himself, whereas Hershey did this with two. Anyway, I won't go into detail about what the experiment was, but I thought, this poor guy, he's just a graduate student, and Crick and Brenner and Watson are ganging up on him. So I went downstairs, and there's this guy sitting there with a big jug of gin and some bottles of tonic and some ice, and he's selling gins and tonics. Frank, he's still one of my best friends. I saw him just a few months ago, and he told me that the reason it was doing that was to meet the interesting people who would accept the offer of a gin and tonic. At the time, I thought it was so he could get enough money to buy more gin and tonic for himself. I don't know what's true. But anyway, I told him about my interest in DNA. He told me about his genetic experiments. He was doing phage genetics at the time. And he was going to come to Caltech that very September to be a postdoc. So we agreed that we would try and make this work together. And it's really, although it may be called a beautiful experiment, it's really two experiments. First, we had to invent the method. And that took all the time. The method of equilibrium density gradient centrifugation. It wasn't as simple as it sounds. There's a whole book about it called The Most Beautiful Experiment in Biology by Frederick Holmes, the Yale historian of science, sadly no longer alive. So if you want to read a real blow-by-blow description every day and all of the frustrations, it's there. But once we had this method, then it was easy to do the experiment. So that's it.
Yeah, fantastic.
Worked like a charm.
Yes. So I know that many a generation of biology student, myself included, were taught, experiments that you and Frank did.
It was very pretty, but did it do any good?
Yeah.
Well, you could say there are two kinds of people, well, really three. There's the kind who didn't even think about DNA. That was the majority. Then there were people who thought there's a mechanism for replication, and in fact, the structure proposed for the gene, in the case of the double helix, was too simple to be true because biology, from that point of view, is very complicated. And nothing in biology, especially the gene, could be that simple. There's another group of people who said anything that simple has to be true. So
Yes.
these two, and these people were all both types were well represented at Woods Hole that summer. So the experiment had a psychological effect because the DNA proposal of Watson and Crick was just a model. It did obey approved bond lengths, bond angles, fine. But there was no other evidence for it. It was very plausible. There was evidence that it was consistent with. And in fact, the double helix was chains round around each other by Francis Crick and Jim Wong, when Jim may have still been at Caltech then, but it was at least 10 years because the crystallography couldn't distinguish left from right. It's a long story, I won't go into it, but my point is that it was just a model. But when people could see these bands that we got in our experiment before their eyes,
Yes.
the right place at the right time and the right amount, exactly as predicted by semi-conservative replication, that had a psychological effect. And in Jim's own book, The Double Helix, he writes that when that came out, he felt a sense of relief. So it had a psychological effect.
Yes.
Now, the DNA molecule had a much bigger psychological effect, unlike any other kind of a molecule. If you look at a molecule of a lipid or a protein, okay, that's nice, there it is. Maybe you could explain a little bit more about what it does, looking at its structure. DNA is completely different. You look at that molecule and it says, here I am. I've got two chains. How do I replicate? Genes replicate. How do I do it? Well, Watson-Crick mechanism. Here I am. I've got DNA bases for them, but they can be arranged in any way. The structure allows any order of bases you want. So that must be a code for proteins. Go figure out how that's done. Here I am. Every once in a while, I switch pieces with another DNA molecule. It's called genetic recombination. Go figure out how that's done. Once in a while, I change. One of my base pairs changes to a different. That's called mutation. So it's like the DNA molecule is sitting there, giving orders. Here's what you should do. And at that time, there weren't that many people working with DNA. And so what you had was usually one or two groups, maybe three, working on problem number one. Like on DNA replication, there was Cy Leventhal and Gunther Stent and Frank and me. That was it.
Yes.
I hope I'm not forgetting anybody, but you didn't have these vast communities. You didn't have meetings with 10,000 people in Miami's largest hotel, nothing like that.
Right.
So it's a very different time. And I'd say it was from about when it really got going around 1955, 56 till 1970. And now times are completely different. I think partly because there was very little, there was intellectual competition, but we wouldn't have thought of sending a manuscript to a journal without sending a preprint to our friends
Right.
to see if they approve the manuscript.
So in some ways, Matt, you are raising an interesting question, and I've heard this said time and time again, that as you pointed out, science in some ways was more collaborative.
Well, I don't know science because that's a big field.
Well, that's true.
My impression is that stereochemistry in those days was very competitive. It was this young group of molecular biologists who had just come into the field, many from physics or chemistry. After all, Francis Crick was a physicist, a certain kind of physicist. Seymour Benzer was a physicist. Dahl was a physicist. I was a physical chemist, so on. So I think that there was a lot of competition in certain other parts of, for an example, one part of chemistry was closely related to industry. And there you have patent law and secrecy and all of that. So no, it was this special new field.
Yes, yes. So, Matt, what brought you to science? Can you think back to when you first realized science was for you, or were you born loving science?
My parents told me that even when I was 2, 3 years old, I'd fool around with wires. And that when I was maybe, I don't know, 3 or 4, I learned that if you touch wires to little bitty batteries and have little lights, you can turn little lights on. I built, when I was a little older, crystal sets. And by the time I was 15, I had a very big laboratory in what should have been the garage of my parents' house, or
Uh-huh.
no longer available as a garage. I was doing rare earth chemistry, and I was doing very fancy stuff. I had a spectrometer, a big lab.
Oh goodness! Wow, wow. So your parents were incredibly supportive.
They were marvelous. After all, well, I don't know if I have time to tell a story.
Yes.
Well, I went to a high school in Los Angeles where I grew up, John Marshall High School. But during the war, is World War II, of course, kids often didn't just vacation. There was a feeling you should do something useful. You could work in a factory, or you could take courses in summer school. I did both. One summer, I factory work in a defense factory. At least one summer, maybe two. I think one. I went to Hollywood High and I took courses in chemistry and math. And then I fulfilled all the requirements under California state law for getting a high school diploma. So I went to the registrar at John Marshall High School and said, I'd like to have my diploma, please. And she said, no, there's a law in California. You must have 3 full years of physical education. Physical education. I said, well, what do I do during that year? Because I needed one more year to add up to 3. She said, well, were there any courses you liked? I said, well, yeah. She said, well, you could take them over again. Well, I thought that was about the most stupid, or not her fault, but the law.
Yes.
Up to that time, I think I had very good relations with my parents and my uncles and aunts. I thought that grown-ups always knew what they were doing. So this just shocked me. And so I started asking, what can I do? And someone told me there was this place, the University of Chicago, that would take you without a high school degree. So that I was 16 when I went there. And when you said my parents must have trusted me, yeah, because this was a town of Al Capone and gangsters and everything, or so we thought. And they let me go all by myself to Chicago, which I loved. And when I got there, I found out you can't study chemistry and physics. All those special courses have been abolished for undergraduates. All you could do is take the great books.
Oh,
And at first I was dismayed by this, but then I began to see this was a wonderful opportunity to do a little bit of reading outside of science and learn a little bit more about the big world.
yes, absolutely. So, Matt, starting with, I mean, you did that fantastic work with Frank Stahl, and you've gone on to do simply amazing work in a variety of fields. But there's one particular area that I think perhaps the biochemists or young biochemists out there might be less familiar with, and I'd love to spend some time on this, particularly because it relates very much to some of the questions and issues that we're facing today. And that's how you got involved with sort of the issues around bioweapons and anthrax specifically. So, I mean, how did you become involved with this?
During the period from about 1963 till maybe 1980, I spent half my time doing what I could call chemical and biological arms control. I
Yes.
don't do that anymore, but it took about half of my time. And here's what happened. One summer, the summer of 1963, Jack Kennedy is still alive. He had created at the State Department a thing called the United States Arms Control and Disarmament Agency. It doesn't exist anymore. But at that time, their job was to think about how to control arms, think about treaties, think about inspections, stuff like that. And they were just started, so they needed new ideas with what to do. Otherwise, they'd have to give the money they had from the Bureau of the Budget back. They had to do something. So they invited 6 academics, I was one of them, to come down there and spend just the summer, but full time. I also had a friend who had a beautiful little house in Georgetown that he wasn't going to be using, and it would be empty all summer long. And I had thought, well, it would only last one summer and it's real fun, and why not? So I went down there and they said, you're going to work on European theater and nuclear arms control. I knew nothing about this. That's okay, because a lot of scientists feel that they can make some progress in any field given a chance, but not if a lot of other people have already been there first. And in this case, Henry Kissinger had already written his great book on this subject. So I went to my boss and said, you're wasting your time and your money on me. Why don't you ask me to work on the control of chemical or biological weapons? Because I know some of that stuff. He said, do anything you want. We're all flying to Moscow next week to negotiate the atmospheric test ban. You can have the desk of a guy named Frank DeRuel. He was doing that, but he got depressed and so he killed himself. But you can take over all of his classified documents in his safe. So I did that. I was very fortunate because I had a little office, only 2 people in the office, and my office mate was Freeman Dyson, the physicist from whom I'd taken my first course in quantum mechanics at Berkeley, and who later became a very good friend. Anyway, so I went to the CIA to see what we thought other countries were doing. And at the time, the answer was, we're not really sure. We had suspicions, but we didn't know anything for sure. And then I went to Fort Detrick, where we were developing our own biological weapons and also defenses, both. And I was taken around by a very nice man, an immunologist, had been a Harvard Medical School faculty, a colonel, I'll think of it. Anyway, we came to a big building about 7 stories high. And when you got up close, you could see that the windows weren't windows, they were camouflage windows. And I asked him what to do, what we do there. And he said, well, we make anthrax spores there. It's a 10,000 liter or 10,000 gallon, I forget, 10,000 something fermenter. I said, well, why do we do that? He said, well, it'll save money. It's cheaper than nuclear weapons, but it's a strategic weapon. Could attack cities with it. I don't think I realized it right then. His name was Fothergill, Leroy Fothergill. But in the taxi ride back from Maryland, where Fort Detrick is, to my office in the State Department, it dawned on me, hey, why would you want a very cheap weapon of mass destruction? Like, for example, what if I could make hydrogen bombs for a nickel each? Oh, you wouldn't want that. Everybody would have some. So it'd be great if war was so expensive that nobody could afford it at all. Okay, no more war, too expensive. And then, being an American, my thinking was, there has to be one country that can afford it. And at the time, I thought of us as a benign country. Later, we got into some wars that weren't so benign, but not before. But this is stupid. This is really stupid. Why would you want to pioneer a weapon that would put mass destructive power in the hands of just about everybody? So I realized that this had to stop. And I also realized another thing, that the argument for it was so powerful that if only you could get that argument in the hands of the person who could stop the whole thing, he would certainly become convinced. There's only one such person, the President of the United States. So like when we write out a protocol for a lab experiment, I wrote out a protocol. And it said, first of all, I have to get to the people around the president. So when he asks people or when they come to see him, this message comes through. I have to also get to the public. There's an old anecdote about President Franklin Roosevelt where someone, a certain banker, went to see him with a certain proposal to which Roosevelt is supposed to have said, I agree with you completely. Now go out and put pressure on me. That it's very more like presidents have lots of things they could or could not do. And it's certainly a lot more easy for them if it's something that has public support. So I went down to the Washington Post. A friend of mine in Cambridge knew the managing editor down there. Howie Simons was his name. And Howie Simons I told him that I needed to get into the newspapers. He took out a pad of yellow paper and he wrote down about 30 names. These were the science reporters at each of the 30 big newspapers. We don't have that anymore. Only a few newspapers have science journalists now. Too bad. But in those days, they could afford it. So I made contact with nearly all of those 30, either seeing them, talking to them on the phone, corresponding with them. And then it got a lot of press because once a week, the editor of the paper wants to have something about science. And this is a jazzy subject, biological weapon. So there were lots of articles. Also, the Army itself cooperated with me because they made a few mistakes. They had a bad accident in Utah, killed a bunch of sheep. They had some leaky tanks, some biological accidents, all of which makes news. And then the other important thing that one would have to do is make allies in the Senate and the House. So I did that. In the House, I became particularly close and cooperating with a congressman named Max McCarthy from Buffalo. And in the Senate, it was Senator Fulbright, the great Senator Fulbright, chairman of the Foreign Relations Committee. And we worked very closely together with the goal of getting this thing stopped. When Henry Kissinger was chosen by President Nixon to be his national security advisor, I knew Henry quite well already because his office was next to the biolabs in the Semitic Museum. And we have lunch together and we had driven around a little bit in Europe together. So I knew him. And well, I guess the driving around in Europe was a little later. But anyway, I knew Henry fairly well. And so once President Nixon appointed him, which was before the president took office, we knew that he had selected Henry for this job. And one day I was coming back from Washington or going to Washington, one or the other, and at the American Airlines, there was a steep ramp down to the gates. And I can't remember whether I was coming up or he was coming up, but we sort of collided like that and kind of hugged. And he said, "Well, Matt, what do you want to do? What do you want me to do about your thing?" Which he knew by then was chemical and biological weapons, because it's a few years after 63. It was after the whole presidency of President Johnson before he got Nixon. So I told him I'd write him some papers. So soon after Henry took office, he got a letter from Mel Laird, the Secretary of Defense, which said that he wanted Henry to have a study done of our policy with regard to chemical and biological weapons. And in this letter, he said, the reason for this is I'm getting an awful lot of complaints and questions from Senator Fulbright and Congressman McCarthy. So in a way, in an indirect way, Laird was responding to what Fulbright and McCarthy and I were hoping would happen. Anyway, so this meant I had a route right to the president. I could write papers for Henry, and Henry would give them to President Nixon. I never met President Nixon. So all the pieces were in place. And then it just had to happen, and it did.
Fantastic. So you've obviously seen how governments respond to crisis, etc. From your perspective, the COVID-19 pandemic that we're all facing now, are there pieces to it? Like, for example, I'd sort of like to get your sense is, what is your sense of both the enormous challenge, but perhaps some of the opportunities that this pandemic really presents for science, and also how science and government work together?
Well, a crisis like this can have either of two opposite effects. The first is that everybody gets together to meet the common threat and suppresses their differences, even differences outside of the actual management of the pandemic, a spirit of cooperation. The opposite is true too. When a crisis appears, it could be that people will start pointing fingers and will not act in a unified, cooperative manner, but in a destructive manner. So it's, I think, up for grabs what we're going to do. It could go either way. But what the pandemic has done is to reveal a lot of shortcomings in the way our system has developed, especially recently. It's not easy to realize unless you look at a graph, but the huge gap between people in the lower half or even the lower 90% and those who have vast amounts of money has just skyrocketed. if you look at the population in the prisons in the United States, it's poking along until around the time of President Clinton. And then it just goes exponentially up. Right now, capitalism has run into some troubles, in my opinion, and partly it's because of technology. Let's imagine that you're the owner of Ford Motors. You're Henry Ford. You get a lot of money, but what do you do with this money? You spend it. You have to buy rubber and steel and paint, and you have to hire a lot of people. You have to build factories. You make jobs with that money. You don't hoard it. You hoard some, of course, and you may collect art and so on. At least that keeps painters busy. But now we have a different spectacle. We have people who are putting vast amounts of money back into the financial system. Now, some of them will give it back in the form of philanthropy, either while they're still alive or after they die, but huge amounts, and in countries, names of people who we don't even know, because huge amounts can be made in the financial market, financial system. So a lot of money is being taken out. Let's take Facebook, for example. I hope I'm right what I'm going to say. I'm only a molecular biologist, but here's Facebook. I don't need big factories. I don't need a lot of rubber, steel, and paint. I don't even need too many employees. I don't need big factories. But I have payment coming into me from billions of people, scraping if they're poor, but not if they're rich, but they're giving me money. So I got this huge amount of money. Now, if I go out and do something with it and employ a lot of people, that's great. And maybe the owner of Facebook, I don't know what he's doing with his money. Maybe he is distributing it. I just don't know. Most of the people who make a huge amount of money this way, not necessarily by being CEOs of a company, but also by their investment procedures, doesn't get back into the system. And so that's very dangerous. It creates a huge gap, and the money isn't being used for creative purposes. Anyway, I may be talking through my hat, but it's clear that in many ways that are outside of what you might think about capitalism or not, how it might need some fine tuning. There's climate change. There's a possibility that, as happened once in the Cretaceous, it destroyed all the dinosaurs. And if it hadn't, by the way, maybe there would have remained little teeny mammals, but not us. I don't think we would have done well with dinosaurs. I don't know. Anyway, so there's the possibility of another meteorite. You can't predict these. There's just an awful lot of them out there. You can see them at a distance. And we do have a program to at least find them and maybe do something about them. What about huge volcanic eruptions that cloud the sky, prevent enough sunlight for growing of crops? And what about pandemics? And what about genetic engineering? Not right now, not next year, not in a couple of decades, but sooner or later, we are going to be guiding our own heredity. We are going to redesign our genomes, at least some subset of humanity is. And that's going to raise very important questions, not so much about what you want to change, in my opinion, but what you want not to change. And very little is known about what's called epistasis. So nearly every protein that's made in a living system interacts at some stage of its functioning with some other protein. And I don't mean just a dimer, I mean a heterodimer. It interacts with a different protein. So now let's say that we want to make a better protein that's structure of the retina. We see better. Good. So you do that. But you didn't know that this same protein, and you get better sight, maybe even that. But you didn't know that this same protein does other functions in which it's binding to something else. The gene product of almost every single gene could, in principle, be interacting with one or more of the products of all the other genes. Now, it's going to be a long time before we figure out all those interactions. If you ever looked at a biochemistry chart with all the, this is nothing compared. Very hard to study. But when we get quantum computers and huge amounts of, maybe then we can do it. But so, in other words, what I'm getting at is there are some things we don't want to change. And what are they? Our humanity, our sense of curiosity, our feeling of awe. It's up to it's a matter for individuals to decide. But how do you do it? How do you make sure you do it? Where is
Right.
humanity in here? Where is awe? Where's
Yes.
curiosity? In other words, we don't know anything about the brain that would enable us to start changing the rest of our genome for other purposes without running the risk of losing something about our humanity. This may sound far-fetched. I'm not talking about bioethics. That's different. That's to prevent loss of your private identity, or maybe would allow unfair discrimination. That's different. I'm talking about the essence of what it is to be human. So biology has a huge future ahead of it if we survive. And we need to have a species point of view, no longer a tribal point of view, or we're just not going to make it. We might make it into some other form. Now, it could even be that the human species now will branch off and we'll get varieties and then subspecies. That can only happen, though, if you have allopatric total separation. But now, Elon Musk says he's going to have people walking around on the moon in 2022. Jeff Bezos is trying for the same thing. Those guys are going to come back, but sooner or later, the idea is to start colonizing first the moon and then who knows, other planets somewhere. So at that time, if there's little enough travel back and forth, then there's a possibility over long, long periods of time for us to split into two separate species, even.
So Matt,
So
you've touched on some really important ways in which the future of science has enormous possibility, but there are also outstanding questions that remain. I'm curious from what you've seen so far in terms of COVID-19 and the pandemic. Has the pandemic changed your opinion of where science's path might end up, or raised some issues that you think are really worth thinking about carefully when we chart the future?
as respect to science,
Science in
just
general,
science.
yes. Or let's say biology, life sciences.
Well, it seems to me that although most young scientists really should just stick to science, I mean, but it's essential that a few, at least, perhaps of the most creative ones, should try to make a contribution to dealing with these species-wide issues, not just the issues from infective disease, that too, of course, but that's within the realm of regular science. But I'm thinking of these other things, trying to understand what is most precious to us. How do we enhance it or protect it? Do we want to have a course at Harvard? We don't have one now on what are called existential threats. Existential threats. If there is such a course, what should it teach, or what should it allow students to learn for themselves? There should be a small trickle of people from science and from good places like MIT and Harvard and Princeton and Yale, all those places, a trickle of minds, think of it in terms of minds, who are entering society, both in terms of public education, but also government, regulation, even industry, who are thinking far ahead about the interests of our
Mhm.
species.
So you're really speaking to sort of a return to or a sustained focus on fundamental issues. And it makes me think that it's been said that as science has become larger and larger, as our understanding of the natural world has grown, it has led to enormous specialization. So, Matt, thank
Yeah.
you so much for spending the time with us. Matt, if you're willing, I'd love to have some additional interviews because I've barely scratched the surface of things that I'd like to ask you. So, but still, thank you so much for being willing to do this.
Thanks a lot.
Okay, take care, Matt.